Right.
Don't worry,
You don't have
you look so
to.
disappointed. So let down. You know, just chat a little bit.
I saw the diagram like that.
So he wasn't.
It isn't. I think it's pretty, so.
Red line.
ENCYSD. Insist. Yeah, you should put that away. Or, okay, you'll get your quiz if you want to.
Yeah, I just put it on.
Don't take a picture.
I'm
Next week we're going to start.
Exclamation point, so students,
Oh, good night. That's supposed to be a 1. H1. Oh, for pity's sake. 1. 1. 1. 1. Okay, for those of you on Zoom, Mrs. Schaefer will have copies of all the quizzes, so please take them either Monday or when you come back. So.
I have a question.
Very good, everyone. That's good, all right. Yeah, we will.
I have a question.
Yeah, go ahead.
Where can I take it?
Do you know where the Natural Sciences office is, Stephanie?
No.
All right, so you come in to Whiteman Gibbs from the east side. So you get into
Oh.
the lobby and you're on the first floor, not the basement floor, the first floor.
Okay.
You walk through there's the big lecture hall 110 to your left. There's the restrooms to your right. You walk down that hallway through the double doors. It's the very first door on the right.
Okay.
And the secretary is Kathleen Schaefer.
Okay.
She will give you the quiz. If you're absolutely lost, can't find it, there's just no hope of finding it, then just come see me. You know where my office is, right?
Yeah, I can find it.
203.
Okay. And then we take it there with her?
Yeah, she will ask you to dump your backpack with her and then surrender your phone, and then she'll give it to you. Just go in the copy room basically and take it.
Okay.
Make
All
sense?
right. Yep. Thank you.
I apologize for the confusion. I sent the Zoom link out because we have some people who are on quarantine because of COVID protocols. Some of you wondered if we were having class today face-to-face, and the Zoom link, I think, caused some of you to think that we might have been meeting via Zoom or not. It's just for the benefit of those who either snowed in or are, okay, I'm kind of a mess today, so don't mind me. Sydney, stop laughing at me. Because there are some people that are still snowed in. My middle daughter lives in Ann Arbor and they got hammered. Took her half an hour yesterday to get out of her driveway. And Dane Osley lives near Saline. It took him 4 hours to dig his car out. His car was just buried in that much snow. It was, yeah, bad. All right, so please have your handout ready. This one. We're going to use that today. So we're talking about DNA replication. We saw that DNA replication requires the unwinding of those strands. And the enzyme that unwinds it is called helicase. Helicase. And on the handout it's called DNA capital B because the helicase is encoded by a gene called DNA B. That's why it's called DNA B in your handout. Now, the topoisomerases go ahead of the helicase and they take out the supercoils because as you get the DNA more supercoiled, that increases the strain on the double helix. It makes separating the strands more difficult. Without that topoisomerase going ahead of it, the helicase eventually will just not be able to unwind the strands. And then once the helicase gets the strands unwound, there are single-stranded DNA binding proteins that bind to the separated single DNA strands to make sure they stay apart and they don't reanneal. And you see those in your handout as the single-stranded DNA binding proteins. Those are given the name capital SSB. SSB. Now, the enzyme that makes DNA is called DNA polymerase. The substrate for DNA polymerase is the deoxyribonucleotides, except it's the triphosphate version of the deoxyribonucleotides. So we say deoxy NTPs, and by this, we mean it's deoxy ATP, it's deoxy GTP, it's deoxy TTP, and deoxy CTP. So instead of writing all that, I can just say this. I like easy, so that's what that means. These are the substrates for DNA polymerase. That's what it will use to make more DNA. Now, as we said before, this enzyme is a sprinter, not a distance runner. It needs to have starting blocks in order to start. And those starting blocks come in the form of small strands of RNA. And the small strands of RNA are called RNA primers. Those are the starting blocks. So if you take DNA with deoxyribonucleotides, deoxytriphospho-ribonucleotides, and you denature the DNA, you give it DNA polymerase, nothing happens. And when people were able to isolate DNA polymerase, they kept trying to get it to work in the test tube and it wouldn't work. And there's some very, very good biochemists who tried to get it to work and it just refused. Then it was clear you needed little bits and pieces of RNA that were complementary to the DNA, and then it worked. And what was discovered in the test tube turned out to be correct in the cell as well. So these small strands of RNA, these RNA primers, they're made by an enzyme that's labeled on your handout as primase. Primase. Primase can make short strands of DNA, of RNA. It just needs a single stranded DNA molecule as a template. That's all it needs. This enzyme can essentially start without starting blocks. It has low processivity, meaning that it doesn't stay stuck to its template for very long. So it makes short stretches of RNA. And these stretches of RNA are 10 to 12 nucleotides long or something like that. They can be a little shorter than that. They can be a little longer than that, but 10 to 12 is a good statistical average. Now, another fact about these polynucleotide polymerases, both DNA polymerase and RNA polymerases, they can only work in one direction. They make DNA from front to back. They can't go back to front. They make it from front to back. So remember we talked about how DNA has a front end, the 5' end. So it goes from 5', it starts at the front and it makes it to 3'. Now this creates a problem. If you look at your handout, which by the way is on the Blackboard site for those of you viewing by Zoom, you'll notice when you unwind your DNA, one of these strands has a free 3' end. The other has a free 5' end. That creates a problem for DNA synthesis for one of those strands. For the strand with a free 3' end, you can make a primer, you can then extend it to the end, and then as the helicase goes up, you're going to need to restart it. For the other end, you can start at the end and then go forward and you can just keep going. So one of these strands will have synthesis that is continuous. The other will have strands where you're constantly having to start and stop and start and start, stop and restart and stop and restart and stop. So the continuously synthesized strand is called the leading strand. The leading strand. And that's indicated here in your handout, the leading strand. Synthesis on the leading strand is continuous. The other strand, where you're constantly having to start and stop and restart and stop and what have you, that's called the lagging strand. The lagging strand. Now in E. coli, the same enzyme synthesizes both the leading and the lagging strand. And this seems to be the case in most gram-negative bacteria. It's not going to be the case in eukaryotes, as we'll see, but this is the case in E. coli. So as we see here, the helicase breaks the hydrogen bonds, separates the strands. Topoisomerase works ahead of the helix to take out supercoils that can slow down the helicase. Single-stranded DNA binding proteins bind to the single-stranded DNA, prevents it from reannealing. Primase makes an RNA primer, and then the DNA polymerase picks it up from there and makes strands of DNA. Now, E. coli has 5 different DNA polymerases, and they're given the very creative names of DNA polymerase 1, 2, 3, 4, and 5. The workhorse during DNA replication in E. coli is DNA polymerase 3. DNA polymerase 3. DNA polymerase 1 also participates during DNA replication, but it works more as an enzyme that fills in gaps. DNA polymerase 1 is also an enzyme that's involved in DNA repair. All right, so these fragments of DNA that have RNA stuck to them as well, those are given the term Okazaki fragments. And once you make an Okazaki fragment, you have a problem. You've got RNA mixed in with your DNA. Is that what you want? No, you want a DNA molecule that's just DNA, because RNA is less stable than DNA. So that RNA needs to be replaced with DNA. So here is a picture showing you your DNA polymerase. You can see DNA polymerase on this strand. This is a leading strand. It makes DNA as fast as the helicase can unwind it. Over here with the lagging strand, we're constantly having to start and stop, and start and stop, and restart and stop, and so on and so forth. So what about these 3 other DNA polymerases? DNA polymerases 2, 4, and 5 are used when the cell undergoes pretty catastrophic DNA damage. So they are air-prone DNA polymerases that don't have high substrate specificity. So they can even bind and replicate very damaged DNA. But that low specificity comes at a price, and that price is they're not as accurate as DNA polymerase 3 or DNA polymerase 1. Yes, McKenna.
So is it just in coli that is all those 5? Or like
Other bacteria also have multiple DNA polymerases. Some have polymerases that are air-prone, others that are less air-prone. They virtually all use the accurate enzymes for routine DNA repair, and they'll only use the air-prone ones for specific types of damage. They're involved in what's called translesional DNA replication. Translesional. Eukaryotic cells also have polymerases just involved in translesional DNA replication. They're used when your DNA really takes a beating. cells have a variety of repair systems for damaged and even broken DNA. What sort of things can break DNA? Any of you know? If you had to guess, what sort of things might put breaks in DNA? Rebecca? Some viruses have the ability, they're called, I love this term. I think this is a great term. Clastogens. A great term. Just the way it sounds. There are some viruses that can cause breaks in DNA. Polyomavirus is known to be a clastogen. Some viruses are not clastogens, but some have clastogenic capacity. So some viruses, sure. What else?
Are you saying like, I could be wrong, like restriction like that?
Restriction endonucleases can, sure. Those are used in the test tube. You're not going to get those inside human cells, but they can if you can get them inside human cells. Not sure why you want them in human cells, but okay, stranger things have happened. You might be thinking of this, the so-called CRISPR technology. It's being considered now for gene therapy. That uses a nuclease called the Cas9 nuclease. Cas9 can put breaks. That would be an enzymatic break you put in DNA. And then it has a system for repairing it. And if you give it a guide RNA with the sequence that you want to use when you repair it, it'll repair it and put in the sequence that you want it to put in. So the problem with it is there's always off-target cleavage that happens. Although people have engineered it so that there's less and less and less. So we might get to the point where it's absolutely perfect someday, maybe. Hard to say. What else can break DNA? How about ionizing radiation? That break DNA? Yes, can. Ionizing radiation. That's why some of you may work radiology, imaging. You're going to have to wear a film badge, and you're going to have to be careful as to how much radiation you're exposed to, because a little bit of radiation, your body deals with it just fine. You have repair systems for that. High exposure to radiation, not good for you at all. When I was in graduate school, we used to use radioactive isotopes for many of our experiments. We had to wear film badges and film rings to determine how much. We had to turn those in every month. 1 month, mine got dumped in the radioactivity dumpster. It fell off my lab coat right in. Like, oh, my film's going to get exposed. So I gloved up and I fished it out of there, and I said, this is not going to be good. So yep, I was exposed. So I had to not use isotopes for a month and so on and so forth. And that was not good. But anyway, ionizing radiation can certain chemicals, things like benzene, things like sorolins, certain types of chemicals that can intercalate in between DNA and react with it. They can break DNA as well. Ultraviolet light can, yes, in high intensity, yes, certainly can. So that's why you should mediate your exposure and wear sunscreen and what have you, particularly if you are of a lighter hue. All right, so repair these other enzymes. These are the ones that are involved in E. coli in DNA replication. Pol3 is a very complex enzyme. It's multi-subunit. Those subunits play a role. 3 of the subunits are involved in the catalytic aspect of it. The rest essentially are for properly proofreading the synthesis of DNA if it makes a mistake. It stops the enzyme, has it go backwards, fix it. This enzyme will make an error essentially one out of every billion times. It's very accurate. Is it perfect? No, but it's very accurate. Hey, 1 out of a billion? I take that. Brianna? No, that tends to cause a kink in the DNA. And that's how repair enzymes actually recognize DNA with a mistake in it. Because the bases aren't matched, so instead of a regular helix, you've got one that has some sort of abnormality in it. They can, yeah. Yeah. You have short patch, long patch repair, single-stranded break repair, double-stranded break repair, air-prone double-stranded break repair, non-air-prone double-stranded break repair. You have nucleotide excision repair, base excision repair. You have several different repair systems. And some types of inherited cancers, for example, are due to mutations in these repair systems, demonstrating the importance of these repair systems, because your DNA is constantly being assaulted. And so it's constantly being repaired. And part of the reason cancer becomes a disease of older age is those errors have started to accumulate in cells because the repair system is very busy. It can get overwhelmed. They can miss things. And so that's why, in fact, the American Gastrological Society has now revised its guidelines. Instead of starting colonoscopies at 50, they've now suggested 45, because it turns out younger people are starting to get more adenomas and colon cancer and things like that. So now 45, that magical age when you have to go in and have a colonoscopy and so on and so forth. And also, as when you get to my age, for example, dermatologists will tell you should have a dermatological screen every year, where the dermatologist looks you over and makes sure that if he or she sees anything suspect, they can deal with it, because tumors are very easy to deal with when they're small and not very advanced. But if they get bigger and more advanced and they start to spread, then they're really hard to deal with. And that's why when you look at the cancers where we have the most success, they're ones that can be screened for, so you get them early. So things like cervical cancers, things like breast cancers now, if they're caught early, they're very treatable, so on and so forth, and what have you. And I'm getting off topic, aren't I? It's your fault. Did you have a follow-up question, it looked like? No? Okay. Yeah. All right, Pol-1 is composed of but a single polypeptide, and it not only removes the RNA primers and the Okazaki fragments, it replaces them with DNA. So this enzyme has a so-called exonuclease activity. Exonuclease activity. So it binds and goes ka-chunk, 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 takes out the RNA, rebinds, puts in the, replaces them with DNA. And that's essentially how those Okazaki fragments get replaced with DNA. That's due to the activity of Pol-1. Yes?
So then what is DNA ligase?
I'm getting to that. So here are some of the subunits that we see in Pol-3. I'm going to show you a video here pretty soon of Pol-3 doing its thing. And you'll see it has a lot of subunits. Each of those subunits plays a role. This beta subunit here is a so-called clamp. There is a ring-shaped clamp that actually clamps around the DNA and feeds it through the enzyme. And that beta clamp is 2 subunits, and they look like a half moon. When they clamp together, they form a donut, essentially. And that donut essentially is around the DNA, and it helps feed the DNA properly into the enzyme. And that's why it's called the clamp loader, which sounds like something from a trucking company. Polymerases, when you look at their crystal structures, you can get polymerases that, in terms of their amino acid sequence, they don't look very similar to each other. But when you look at their crystal structure, all of them have this very similar structure where they look like a hand. So they have a thumb, finger, and a palm domain. So they all kind of look like, and again, you can get enzymes from things as far flung as some sort of invertebrate that lives in hot springs, to algae, to bacteria, to yeast, to fish, to frogs, to people named Fred, to whatever. All of their DNA polymerases and RNA polymerases too. Their crystal structures have this hand sort of look. So they have a so-called thumb, finger, and palm domain. The palm domain is the catalytic domain. And you can see that the finger and the thumb kind of hold the double helix as it comes into the molecule. Now, it turns out the finger and the thumb domain, that helix, if it comes out, if there is something awry in that helix, if you have bases that aren't well matched, the finger and the thumb domain detect it, the enzyme stops and it works backwards and it gets rid of those errant bases and then it restarts and it puts it in. So the finger and the thumb domain are part of the proofreading function of the enzyme. And you can engineer enzymes that have kind of stubby finger and thumb domains. They make a lot more mistakes because the finger and the thumb domain are the thing that detect whether or not the helix has some sort of structural abnormality in it. And that's essentially is one of the things that makes these enzymes so accurate. All right, so as we said, it can't start by itself. It needs RNA primers. It has to go in only one direction, 5' to 3'. And so you have at the replication fork, you have replication occurring in two different directions. And this essentially reiterates what we have from the handout here, where with one strand, your synthesis is going to be continuous. That's the leading or lagging strand. Leading strand, the other strand, you will need to constantly start and stop. That is the lagging strand. And that will give you two molecules with one strand that is from the former DNA molecule, the other that has been newly synthesized. So the leading strand is only going to have one RNA primer. It only needs one. You start with one and you take off. Whereas the lagging strand will have multiple RNA primers. So it's a strand that will have the main Okazaki fragments. And as you can see, these Okazaki fragments are quite long in bacteria, not quite as long in eukaryotes. They're up to thousands of nucleotides long in bacteria, where they're only a few hundred bases long in eukaryotes. And then Pol 1 binds to the Okazaki fragment and chips them out. And then once it chips them out, then it puts the bases in. But once it gets to the end of the other molecule, it can't seal the nick in the backbone. You need a special enzyme to seal the nick in the backbone. That enzyme is called DNA ligase. DNA ligase. And so this slide essentially recapitulates much of what we've said. Remember, DNA replication is bidirectional. So I've drawn for you on the handout essentially what a bidirectional molecule looks like. You'll see that you have leading strand, lagging strand. The other side is kind of a flipped mirror of that. Instead of leading, lagging, now it goes leading, lagging, like that. Where it was leading, lagging, now it's lagging, leading on either side. So both in bacteria and in eukaryotes, your origins of replication initiate replication in two directions. So you have bidirectional replication, just as you see here. So now this concentrates just on the one side. So over here, that's the leading strand. Over here, you have the lagging strand. As the helicase continues to unwind that DNA, you make another primer, replicate it. Make another primer, replicate it. Make another primer, replicate it. Paul 1 chips these out, replaces them, DNA ligase seals the negative. Chips those out, DNA ligase seals it in. Paul 1 chips it out, replaces it, DNA ligase seals it in, and so on and so forth. And there it goes. And this essentially is meant to show you essentially what you have on your handout. So when you have the helicase and the primase working together, and it's clear that the primase interacts pretty strongly with the helicase. So the primase kind of follows the helicase along. So it follows it, makes a primer. Keeps going, follows it, makes a primer. Follows it, makes a primer. And that complex between the helicase and the primase is called the primosome. Primosome. And then the primosome itself is also associated physically with the DNA polymerase, and that complex with the helicase, primase, and Pol III is called a replicosome or replisome. Replisome. All right, let me show you what this looks like in a video. So I don't think I had the right one there. Oh, here we go. All right. Okay. Okay, yeah, these wretched commercials. Okay, we just let that play through and then skip. Okay, stop. All right. Okay, so let's get that so everyone can see it. All right, and this has an animation. This has a narration. So if we plug in this, we should be able to hear the narration as well.
Replication. Both strands of the double helix act as templates for the formation of new.
There.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs in a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is immediately at the top of the frame, and the other new strand. The first step. The lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then When the copy is complete, each section is released and the next is drawn back for replication. Intricate as this mechanism is, the exposed strands of single DNA are covered by
Okay. That is definitely one of the best animations of DNA replication I've ever seen. It is a extremely precise, well-done animation. Again, that animation is on Blackboard. If you want to watch it, there's a more extended one that also includes transcription here. There's also an animation about chromatin, if you are interested in that animation as well, also very, very well done. Okay. All right. Okay, so the lagging strand, as you see, is one where you're making the Okazaki fragments and the polymerase synthesizing the lagging strand also moves towards a replication fork, but it's replicating DNA in a different direction. Once it gets completed, essentially, it has to just dissociate and then let the DNA go and then rebind it, essentially. And that's how you get that phenomenon going over and over and over again, just like this. And so the two catalytic sites, as you can see there, one of them is making the leading strand, the other is making the lagging strand. And the primases are with the helicase, and then you have the clamp loader, and then topoisomerase is working ahead of it, and so on and so forth. Termination of replication occurs, at least in bacteria, once you get all the way around the chromosome. The sites for termination are called TER sites. TER sites, and there's two of them. There's a so-called T1 and a so-called T2. And if the T1 site is bypassed, it will stop at T2. If the T2 site is bypassed, it'll stop at T1. There's a protein that binds to the TER sites called termination utilization substance, or TUS. This is a protein that binds and prevents DNA replication from proceeding any further, essentially. So you get essentially the molecule being replicated like this. Here's the T1 site, here's the T2 site, and the replication can't proceed past TER2, but it can't go past TER1. Replication come the other way, can pass T1, but it can't pass through T2. Once it gets to this site, essentially, the two molecules have been properly replicated, but they might end up being interlocked. If they're interlocked, they form something called a catenane. These catenanes can be resolved by topoisomerase. And topoisomerase essentially will untangle them, and now you have separate molecules. Now, it's worth mentioning that bacterial chromosomes are also attached to the membrane. That attachment to the membrane is important in its regulation of when it can initiate transcription. It's bound to the membrane by means of a protein called SEC, S-E-Q-A, SEC-A. And SEC-A keeps it bound to the membrane, and every time you look at bacterial chromosomes, they're associated with the membrane in some way. That is something that's been known for quite a while. Any questions about DNA replication in bacteria before we go on to eukaryotes, like humans? Well, replication in eukaryotes is very similar. Mechanistically, it's virtually the same as it occurs in bacteria. Having said that, the proteins that are involved, while they do the same thing, the proteins are very different. So if you were to take the two, take for example, the clamp loader complex in eukaryotes, the protein that makes the clamp loader forms a donut, but it consists of 3 subunits, not 2. And the amino acid sequence of that protein is not at all similar to the sequence of the protein in prokaryotes. Having said that, they do exactly the same thing. So these proteins are so-called analogous proteins, but not homologous proteins. They're analogous, but not homologous. In eukaryotes, remember, you're replicating linear molecules, not circular molecules. You have to deal with chromatin. So the chromatin has to be removed after you replicate DNA and then reassembled back onto the DNA. And you only replicate the DNA during a specific time of the eukaryotic cell cycle. So to refresh your memory, from the very beginning of A&P and then pathophys last year, which I know was a very long time ago, that's why I'm refreshing your memory. There is something called the cell cycle that eukaryotes go through. You'll remember the cell cycle consists of those stages: interphase, prophase, metaphase, anaphase, telophase. You learned in high school, those are the stages of mitosis, right? So the cell cycle includes mitosis actually refers to nuclear division. The actual division of the cell is called cytokinesis. Now, these are coordinated processes, but it's been shown in the laboratory you can separate them. So cytokinesis is now regarded as a separate process. Mitosis refers more to nuclear division. And if the cell cycle was 24 hours long, just to pick a number, 1 hour of that time would be spent in mitosis. The rest of the time is going to be spent in interphase. So all this is interphase. An interphase is broken into 3 phases. There's a phase called G1. And I can't really divine whether G stands for growth or gap. Because some papers say it's a gap phase. That's why it's called G. Others say, oh, it's growth because the cell is growing. It's G1. Gap or growth, I don't care, just G1. The other phase is called S phase. Now this stands for synthesis. Synthesis of what? DNA. This is the only time during the cell cycle when you can replicate your DNA. Then there's a third phase called G2. Gap or growth? I don't know. One or the other. Pick one. And now it's readying the cell for mitosis. So whereas bacteria replicate their DNA throughout their cell cycle, eukaryotes restrict DNA replication to one window within interphase called the S phase. And you have a bigger genome to replicate, don't you? So eukaryotic chromosomes have multiple sites where you can initiate DNA replication. And because you're doing it kind of all at once, you'll be able to replicate essentially whole chromosomes really quite effectively and really rather quickly. And again, this was demonstrated by Huber and Riggs back in 1968. They also demonstrated bidirectionally. DNA proceeded, replication proceeded bidirectionally from these sites of replication. And they used this procedure called autoradiography. So essentially by pulsing radio-labeled nucleotides or radio-labeled nucleosides, they used radioactive carbon, not radioactive phosphorus. Radioactive phosphorus is too energetic. If you use that for autoradiography, you'll get a smear. So they use C14 labeled nucleosides. The cell will phosphorylate them and use them. And as you radio label them, you can see the origin of replication is here. You're getting replication in that direction. And as you incubate this with radioactive nucleotides longer and longer, this now starts to become longer and longer and longer and longer, demonstrating the bidirectional nature of replication. Also, you see many of these across your chromosome spread, showing that there are multiple sites where DNA replication occurs. And today we know those sequences are called autonomously replicating sequences, ARSs. We know the sequence of them, and you can essentially insert them into bits of DNA you want to put into cells if you want them to be properly replicated. And so you get multiple so-called replication bubbles. Those bubbles then eventually fuse, and then you have replicated chromosomes, and these are now called sister chromatids. Sister chromatids. So the first autonomously replicating sequences were first identified by David Botstein at Caltech in the 1990s, 80s, in the 1980s. And in the yeast, they're 50 base pairs long, but they have this core sequence that you can see is very AT rich. The origins of replication in mammals, they're not as well defined because they vary quite a bit more. They occur at various sites throughout the chromosome. It's also clear in eukaryotes that these origins of replication, they initiate replication at different times during S phase. Some are initiated early, some are initiated in the middle, some are initiated late. Why is that significant? Because you have in eukaryotic chromosomes something called fragile sites. Fragile sites are areas where you're more likely to get chromosome breaks. And it turns out these fragile sites correlate to areas where you have origins of replication that fire late. So have any of you heard of Fragile X syndrome? If you work peds or family medicine, you will see Fragile X syndrome. Besides Down syndrome, it's the #1 cause of inherited mental challenges, mental disability. That's the term that's used now. So Down syndrome would be the #1 inherited cause of mental disability, intellectual disability. Fragile X syndrome is the #2 cause. It's due to a break at the tip of the X chromosome. That site is a fragile site. It's a site where you have a late firing origin of replication. Sydney, I don't like that look. You're looking lost. Sorry? A little bit lost? Okay, ask a question then. Let me back up. Okay, that? You initiate at multiple sites. Those sites then eventually fuse. When they all fuse, you end up with 2 separate chromosomes. Both are double-stranded DNA molecules. They're still bound together though, and they're called sister chromatids. All right. Those sites, they don't all fire at the same time. Some fire early, some fire in the middle, some fire late. And those ones that fire late are more prone to chromosome breakage. Because if DNA replication gets delayed, you have a stretch of single-stranded DNA. Is that as structurally strong as a double-stranded? No. That's why they are fragile sites. That help? Okay. When you get lost, you zone out, and I can tell. You're not a very good liar, are you? Not bad, actually? Oh, okay. So I shouldn't play poker with you. Now, there's been a lot of work, and I don't want to belabor this too much. Just suffice it to say, in order for an origin of replication to be identified, it has proteins that bind to it. Those proteins are going to invite other proteins to bind. And there's this protein complex. It's called the origin replication complex, or ORC. Since I'm a Lord of the Rings fan, I think ORC is a really cool name for protein. ORC turns out to be a complex of 6 proteins that are all the same. And they sit on these origins of replication. It was thought that they only assemble during S phase. Turns out they assemble really early. They assemble as soon as a cell gets out of that phase. So these essentially are a sign on the DNA, hey cell, hello, I'm an origin of replication. That's what the ORC complex does. The origin recognition complex, that's what it does, the ORC. Now, later on during S phase, the helicase protein is made. And the helicase protein is called MCM. Now, MCM is 7 proteins, and they're all different proteins. So they're called MCM 1, 2, 3, 4, 5, 6, 7. And they bind and they make this ring. So they bind, so the ORC complex is like this, and they bind here and they make a ring around the DNA, and they will gradually start to unwind it. Now, here's the thing about origins of replication. So let's say you take a frog oocyte. You fuse this frog oocyte with a frog skin cell. That's actually pretty easy to do. You can treat it with polyethylene glycol. These two will fuse together. And then you end up with a frog oocyte now that has 2 nuclei in it like this. If you stimulate this with progesterone, this cell now will start to divide. So will this nucleus start to divide? But guess what? Divides once and only once. That tells you that these origins of replication, they need some sort of passport, like a vaccine passport, to replicate. We call these proteins licensing factors. Licensing factors. And it turns out there are proteins that are in the cytoplasm. They get into the nucleus. They say, okay, go ahead and replicate. After they say go ahead and replicate, they're degraded. So this will replicate as DNA. Only does it once because it doesn't have the licensing factors. This guarantees that this origin of replication, it replicates once during the cell cycle and only once. Because if you replicate it again, you're going to have a mess, aren't you? You're going to have more than one chromosome. That gives you a condition called aneuploidy. That's a term that was in your pathophys book. Although it's a term you've probably long since forgotten. Does anyone remember what aneuploidy means? Macy, I see smoke rising.
I don't know it.
It doesn't ring a bell at all?
No, it does. I read it like last week.
Yeah,
in the book.
in the book, and yeah, so, but it's just, okay, like the Gospel of John that I memorized last year, I was really having trouble remembering it.
Yeah.
Caitlin, how many chromosomes do human cells have? 23. Good. We call that condition euploidy. Euploidy. That means you have the normal number of chromosomes. If you have too many or too few chromosomes, you have aneuploidy. Aneuploidy is not conducive to life. Not conducive to life. Many cancer cells are aneuploid. Okay, that brings us to 2:30. We will pick this up on Monday. We'll talk about eukaryotic replication and some of the players, and then that will take us into transcription, and then protein synthesis, and the first test. Okay. That's it for today, folks on Zoom. I will see you guys on Friday. On Monday.
Thank you.
You can tell it's Friday for me.
Thank you.
Thank you.
I